Welcome to The Explainer. Today, we're stepping into, well, the ultimate forensic crime scene. But look, we aren't dusting for fingerprints on a weapon or looking for muddy footprints, right? We're looking deep inside the human body, literally straight into the DNA of cancer cells. The complex world of the cancer genome is this bustling, chaotic environment, but it is far from random. It holds the actual secrets to how a tumor was born, how it grew, and most importantly, how we can stop it. So, can we actually read tumors' past? It's a pretty provocative question, isn't it? But the short answer is absolutely yes. Tumors, they don't just magically appear out of thin air. They're the result of accumulated damage over a long period of time. And every single time a cell's DNA is damaged and then improperly repaired, it leaves behind a physical diary of its destructive history. By reading this diary, we can reconstruct the exact events that led to the disease. To get there, we've got a clear roadmap for this explainer. First, the genetic crime scene, followed by the usual suspects. Then we'll look at the inside job, the master key, and finally, the future of cancer care. We're going to break down this complex biology step by step because, honestly, the payoff in modern medicine here is massive. Okay, let's dive into this. Let's get our vocabulary straight right out of the gate. What exactly are we looking for at this crime scene? We're looking for SBS signatures, or single base substitution signatures. Think of these as distinct, recognizable patterns of mutations. They're literally the scars or the fingerprints left on the DNA by very specific, damaging processes. Whenever a single letter of the genetic code is swapped out for another, basically a typo in the DNA, it happens in a recognizable context. All right, Section 1, the genetic crime scene. Let's see how scientists categorize these fingerprints. So, scientists are basically acting as molecular detectives here, meticulously categorizing these genetic fingerprints. And to give you a sense of the sheer scale of our genomic library, they've actually cataloged 107 distinct reference signatures in the massive cosmic database. That is 107 unique ways our DNA can be systematically damaged and scarred. Some of these are super common, found across many types of cancer, while others are incredibly rare, showing up in just a tiny fraction of a percent of tumors. But every single one, it tells a very specific story. Which brings us to Section 2, the usual suspects. These are the external, everyday forces that attack our cells. So who exactly is leaving these 107 fingerprints behind? Well, specific lifestyle factors and environments map perfectly to exact mutational codes. For example, if we spot the signature SBS7, boom, we know exactly who the culprit is. Ultraviolet radiation, sunlight. It leaves a very specific mutation pattern that we overwhelmingly find in melanomas. What about SBS92? That is the unmistakable, undeniable fingerprint of tobacco smoking. And then there's SBS1. This one is fascinating, actually, because the culprit is, well, it's just the simple passage of time. It's an aging clock-like signature caused by the spontaneous breakdown of molecules in our DNA as we simply get older. But here's the really cool part. These fingerprints do way more than just identify the culprit. They actually act like a biological clock. When we look at the evolutionary history of, say, a melanoma, that UV signature, SBS7, is found right there in the earliest initiating events. It's the primary damage that kicks the whole thing off. Then, as the tumor evolves, it accumulates these subclonal mutations. And finally, in the late stages, completely different signatures start to emerge, like SBS38, which acts as a late clonal event. Reading these signatures literally lets us rewind the tape and watch the tumor evolve over time. It's wild. Moving on to Section 3, the inside job. What happens when it's an internal system failure? Because, not all damage comes from the outside worlds like sunlight or tobacco. What happens when the cell's own repair machinery just totally breaks down? Now, what's really interesting here is that we all have genes specifically designed to fix those DNA typos we just talked about. MBD4 is one of them. It acts as a base excision repair mechanism. But if a tumor has an internal defect, specifically a biallelic inactivation of MBD4, that repair pathway completely fails. The result? It creates this absolutely pure, hypermutated fingerprint known as SBS96. We see this specific internal failure heavily in certain cancers like UVL melanomas of the eye. And MBD4 isn't the only internal system that can go offline. Imagine your cell's DNA replication system has its own internal spell checker. That's DNA polymerase delta, or POLD1. If that spell checker gets mutated, say with the specific POLD1 R817W mutation, it just starts making rapid-fire mistakes. It misincorporates nucleotides, essentially printing the wrong letters right into the DNA. Actually, scratch that. It's exactly like a broken keyboard.
This specific internal failure leaves its own unique trail of typos, creating a super rare fingerprint known as the SBS 105 signature. This brings us to Section 4, the master key. This is where we talk about precision oncology.
So we can identify environmental damage, and we can spot internal cellular repair failures. But so what? Why does mapping all of this matter so much? Well, let's contrast a standard tumor with a highly specific variant to see how internal defects change the treatment landscape entirely. A standard tumor has average mutation rates and responds to standard treatments. But variants in the SMC56 complex, which, by the way, is crucial for chromosome maintenance, they trigger an absolute cascade of chaos. They cause pole proofreading dysfunction and mismatch repair deficiency, or MMRD for short. This results in a massively elevated tumor mutational burden. I mean, the tumor is absolutely riddled with mutations.
And this brilliantly illustrates why that chaos is actually an incredible opportunity. You might think a tumor with a massive mutational burden is a straight-up death sentence, but the survival data reveals the exact opposite. No, really. Patients with these specific SMC5/6 variant signatures, the ones driving that pull and MMR dysfunction, actually have a vastly superior survival probability. Why? Because tumors with this specific mutational signature are exceptionally vulnerable to immunotherapy. By reading the signature, doctors know exactly which weapon to pull from the arsenal.
Finally, Section 5, the future of cancer care. Let's zoom out and look at the big picture.
Let's summarize the key takeaways that we should all have from this explainer. First, genetic mutations are not just random static. They are physical evidence left at a crime scene. Second, grouping these mutations into SBS signatures reveals the exact cause or the etiology of the tumor, whether that's UV light, tobacco, or a broken internal MBD4 repair gene. And third, knowing the signature unlocks the cure. It points us directly toward precision therapies, like using immunotherapy for those high mutational burden tumors driven by SMC5/6 variants. Literally, reading a tumor's past is the ultimate tool for saving a patient's future. So, I want to leave you with this final provocative thought. For a century, we've basically treated cancer based on its zip code, right? Lung cancer, breast cancer, skin cancer. But what if the future of medicine means we stop treating cancer based on where it lives in the body and start treating it based on the unique mutational signature it leaves behind? That is the true promise of the Cancer Genome Detective, and it's already reshaping the world of precision oncology as we know it. Thank you so much for joining me on this explainer, and keep questioning the science.